So welcome everyone for the third part, which is gonna be now the real thing, the selection. How do we detect selection? Because as I said, like the new hypothesis is that most of the variation we see is mostly about demography and stochasticity. So we cannot just measure things at the frequencies and just think it's about selection, because most probably it's not. But so we've been through this, so now we can really move into selection. So usually when we think of selection, we think it in a comparative way. We're thinking that, okay, there is some selective pressure acting on lineage B, let's say, which is not acting on lineage A. And because of that, we're gonna be able to compare this to lineage, and the difference we can see there might be due to selection. And we think of selection and there are different types of selection, let's say, but here are three main that we're gonna talk about, mostly, of course, positive selection. So negative and or purifying selection is selection that will remove any new mutation, any new derived mutation, which, as you can imagine, means that most of new mutations are under purifying selection, as we already discussed. Then, as I said, we do this in a comparative way. So we're looking at what's going on in one lineage under selection, and we compare it to an outer or sister lineage. And this gonna indicate us what sort of selection we are facing. So if we are facing negative or purifying selection, the consequence in terms of genetic variability at the specific locus or gene that is under this selection is reduced diversity. And why reduced diversity? Again, it's because selection will act against any new mutation. So if no new mutation can be maintained in the population, that means what? That means that it's going to be mostly no variability. And here I wrote about Tajimasdi. The reason is because as most of the tools that we've seen with which we can measure demography or stochasticity, we can actually use the exact same tools to look at selection. And why? Because if we look at the whole genome, we can assume that the whole genome or neutral places of the genome can tell us about demography. But if we look at a specific gene, then it could well be that allele frequencies at these specific genes are behaving differently from the rest of the genome. And that might indicate that actually we are watching some selection going on. So keep this in mind. Tajimasdi and other tools actually can be used in many different ways, either to infer selection at specific loci, or if we look again at the whole genome, to infer demographic events. So here in this case, at this specific gene, we're going to have negative Tajimasdis, indicating again that the observed genetic diversity at this gene is lower than what is expected according to the population size. Now, if we compare this lineage B, let's say, to lineage A, usually what we're going to see is that the genetic divergence between lineage A and B at this specific gene is reduced. And why it's reduced? Because purifying selection acts against new mutations. That means what? That means that the ancestral allele will be maintained. And most probably, this ancestral allele is also present in lineage A. That's a parsimony. So that means what? That means that most probably genetic diversity is reduced, but also divergence is reduced because both lineages will share the same ancestral allele. And when it comes to the side frequency spectrum, that means what? That means that the new alleles, new mutations that might still happen in this lineage B will for sure be very rare. They will be on the left side of the side frequency spectrum. Okay. Next one, positive selection, which is one that's gonna interest us mostly, of course. In adaptomics, it's usually what we're looking at. So in this case, when we look at genetic diversity within the lineage, we're also gonna see reduced diversity. And why? Because now it's a bit different scenario. It's simply that if an allele is beneficial enough, it will have a strong impact, positive impact on the fitness, which means that it's going to spread in the population because individuals carrying this allele will have a strong advantage compared to the others. And because of that, at this specific gene, again, genetic diversity will be reduced because most probably the allele will be fixed. The beneficial allele will be fixed. So there won't be much other alleles present in the population to reduce genetic diversity. And again, that goes with a negative Tachimaz-D, again, locally. Like what I mean by locally is on this gene, but not in the rest of the genome necessarily. Because the rest of the genome indicates us about demographic events. Okay, now when it comes to the divergence, this time we're gonna have the opposite scenario. We're gonna have actually quite increased divergence. And the reason for that is because we assume, or the scenario is that in lineage B, we have a specific selective pressure, whatever. The lineage B is living on a harsh environment and not lineage A, okay? And because of that, that means that mutations that are giving an advantage in a harsh environment, let's say, have no reason to be fixed or to spread in the lineage A because lineage A doesn't need these alleles, okay? So simply that means what? That means that new mutations, new alleles that will spread and get fixed in lineage B will probably not be found in lineage A or found in very rare frequency. And because of that, again, at this specific gene, you're gonna have increased divergence between lineage A and lineage B. And also, as I already mentioned, on the side frequency spectrum, usually the sites, the mutations, new mutations that are under positive selection will be found on the right side of the spectrum. They will be found with high frequencies, and eventually they will be fixed because, as I said, if they give an advantage to the individuals carrying these alleles, they will simply be favored, and eventually the allele will get fixed in the population. And then there is this third type of selection that we won't talk much, but I just want to mention it, is balancing selection. Balancing selection or diversifying selection is a type of selection that actually favor the maintenance of several different alleles, maintenance of genetic diversity at a specific spot. So that's, for example, the case in plants with the innate immune system. The innate immune system genes are usually under diversifying selection, it has been found. And the reason for that is probably that it is more advantageous for a population to carry several different alleles so that the population can resist or let's say adapt better to any sort of pathogen. So that's one case of diversifying selection, for example. Okay, so of course, as I just said, precisely diversified selection increased genetic diversity at a specific site, so we're probably gonna see positive Tachimaz-D. So about the divergence with lineage A and B, then this time, well, it's gonna be rather reduced. And why reduced? Because if there is diversifying selection, there is increased diversity, as I said, which means that most likely, because by chance, if there is increased diversity of alleles, that means that the alleles found in lineage B are likely to also be found in lineage A. Simply by chance, if you have, whatever, on one SNP you have 4 bases, A, T, G, C, and diversifying selection maintain 4 different alleles, A, T, G, C, then of course, lineage A will also have this allele. So in that sense, divergence will probably be quite low between A and B at this locus under diversifying selection. And then finally, about the frequency of new mutation, then simply there won't be any fixation, as I said, because precisely diversifying selection acts against the fixation or the disappearance of alleles in general. So it means that many alleles will probably be at intermediate frequencies and relatively equal frequencies with each other. Okay, so let's move on to what interests us in adaptomics, which is positive selection. And this is a school case, so that's what we call the hard sweep, a hard selective sweep. What does that mean? That simply means that the allele will be so advantageous that eventually it will get fixed, as I mentioned before. And the way you can imagine it is, okay, you have here new mutations, like new red mutation is giving an advantage, and the blue ones are neutral, okay? And across time, so each line is an individual, and you see that across time, then the more and more individual carries this advantageous allele until at some point the red allele will get fixed, which leads again to low genetic diversity at this specific site, because get fixed, no other allele is existing in this population. But what you can also see is actually that the blue ones here that are neutral have no reason to be fixed because they don't bring an advantage, right? But guess what? What's happening here is that one allele is not simply segregating alone. Most of the time, as we already discussed, there is genetic linkage between closely related or closely located, let's say, alleles, okay? So because there is no recombination, that means what? That means that between them, because recombination is a function of the distance, so the closer two alleles are, the less chance there is to have recombination between them, which means the more chance they have to be inherited together. So here what is happening is that because of no recombination, so genetic linkage, not only just the allele that is beneficial, but also all the alleles around it will also be selected and inherited together with the beneficial allele. So that means that this reduction of genetic diversity does not affect only the advantageous allele, but also actually all the alleles or all the loci around it. And of course, the further we go, then we start to get recombination. So then again, we start to get a recovery of genetic diversity the further we go from this selected site. And that's how it looks like. So that's more or less the same idea. So here we have the selected site, so the sweep, we call the sweep. And we have measures of Tadjimazdi, the linkage, the equilibrium, and variability, okay? So all these three gives more or less the same information, which is, as I just said, reduce the genetic diversity because this allele gets more or less fixed, the advantageous allele, the advantageous mutation, but not only this mutation, but all the mutations around. And you see that the further we go from this specific mutation, of course, the lower the linkage, so the lower the probability of being inherited together is. Okay, so linkage is going down until it reaches finally like so let's say that until it reaches the genome average, let's say. And now if we look at Tadjimazdi, so as I told you, Tadjimazdi and the tools can be used in different ways. They can be used to measure demographic events if we look at the whole genome, but they can be used also to measure selection if we apply it locally. But here we need to be very careful because if you only measure Tadjimazdi at this position, you're gonna get whatever, a negative Tadjimazdi. The problem is that if you get just one position, Tadjimazdi is negative. This can be explained by two things. It can be explained by a population which is in recovery. Remember that when the population is recovering, then there is a lot of individuals, but the genetic diversity is pretty low because the new mutations didn't have time to arise and to accumulate while individuals are being able to reproduce. So if we have at one spot negative Tadjimazdi, it's either a population being in recovery or it can be some sort of selection happening. But if we have only just this information, we can only conclude one thing. This is most probably due to demographic events or population being in recovery, okay? Because that's, as I said, it's a paradigm that most of the genetic variation or so we observe is due to demographic events. So having the Tadjimazdi at your favorite gene is not helping anything. The only way that you can tell if selection or not is happening is by comparing locally how Tadjimazdi behave with the rest of the genome. The rest of the genome will tell you about demographic events because it's just neutral, most of it. And that gives you the background, that gives you the demographic history. And if your gene behave differently, then okay, then you can imagine that something is happening, which is the case here, for example. So you see that Tadjimazdi is there and for the rest of the genome, but when we look at the specific selected sweep, then suddenly Tadjimazdi drops. So there is lower genetic diversity at the specific selected position. That makes sense with the idea again that the allele get fixed. Okay, so keep this in mind. That is really, really important. And that's what the author did here. For example, this is like a concrete example. So what I did is looking at the gene that is involved in UV protection, and they look at Tadjimazdi in that gene in people from African descent or European descent. So what they see here is for European descent, they see negative Tadjimazdi, suggesting that there could be selection. But of course, they can again only conclude this by comparing this to the rest of the genome. You see that most of the rest, I mean, the rest of the genome is actually having a positive Taji-D, suggesting that humans are still not recovering in terms of population, but are still affected by the bottleneck. Okay, we are just after the bottleneck. And again, the author could conclude that there is selection going on only by comparing what's going on in this specific gene versus the rest of the genome. Because again, if you just would take this part here, then no idea if it's about demographic events. So again, population being in recovery, or if it's really about this gene being under selection. And especially by comparing it with people from African descent versus European descent, then you can start to think about what it means. You remember, like when we look at selection, we usually do it in a comparative way of one lineage where we expect a certain selective pressure versus one sister lineage where we don't expect the selective pressure to happen. So this comparative way is also very important that I'm gonna go on with now. And that's what we have in here. So I've already talked about it, but let's recap that. When we want to look at selection, we usually do it in a comparative way of one lineage undergoing certain selective pressure and the sister lineage not undergoing this selective pressure. And once we have this, we do two things. As I showed you in this table, we measure first the diversity within the lineage ongoing selection. Here it's human. And we measure the divergence between our lineage of interest and the sister lineage. And by comparing that diversity within versus divergence between, then that will tell us something. And again, if we look at positive selection, we expect very low diversity at the selected sites. And why? Again, because the advantageous alleles will spread in the population and will get fixed. So no diversity ever. On the other hand, when you look at the divergence, this time, these mutations that are selected in lineage 1 are probably not selected in lineage 2 because that's precisely why we are comparing. We are comparing with a lineage where we don't expect this selective pressure to happen. So that means what? That means that these new mutations that arose in species 1 and spread and are fixed will probably not be found in lineage 2. And that means that the divergence between the two lineages is going to be super high. So we can take the example here. You see that there are 3 mutations here that have been selected probably because the diversity is low, they are fixed. These 3 mutations are simply fixed in humans. But when you look at chimpanzees, then you see that this mutation is simply not found. While this mutation here is not the case, you see that it's not fixed to start with, and also it's also found in the chimpanzee. And then here's the same, typically. And of course, all these white ones, not the non-derived mutations. But if we look at neutral, you see that there is simply no pattern. Like there is a bit of diversity or relatively high diversity, and sometimes it's found. If we find that this new mutation is also found in chimpanzee, sometimes not. So you see that in neutral case, you see that there is simply no pattern whatsoever. And now if we compare diversity versus divergence, so again, diversity within one lineage versus divergence between the two lineages, we're gonna see something like that. We're gonna see that at this position, which is a selected position, selected site, we're gonna have a drop of this ratio. Because why? Again, because diversity is gonna be low, because the allele is fixed or close to be fixed. But divergence will be super high because, again, one lineage is under selective pressure, not the other, so it gets fixed in only one lineage. So if you make a ratio of the two, that gives you what? That gives you a very low value because low diversity divided by high divergence, it gives you a low value. And again, this has to be compared with the rest of the genome. Otherwise, if we would see the same value for this position, but also the rest of the genome, that tells us something else. That tells us that low diversity, so our lineage is experiencing extreme bottleneck, for example, and high divergence could simply be because divergence is super high between the lineage. We chose a lineage that is very far away from the other. So always remember that we need to compare to be sure or to see that we have selection acting on a specific site. We cannot just look at the value itself, the absolute value. We have to compare it with background, with the rest of the genome. Otherwise, you cannot disentangle demographic aspects or selective processes. And this position here could well match either this one here, this one there, or this one there. So one of these three here that are probably been selected. And if we continue, there is one way, especially one precise tool that people use quite often to compare, again, the exact same thing, to compare intra-lineage genetic variation versus comparing divergence between lineages that we are comparing. And this is FST. And we do FST scans. What does that mean? That means that we take windows in the genome, okay? And then for each of this window, we scan, which means that we calculate the FST, and then we see what we can see. So again, it's the exact same story, okay? So comparing intralineage variation versus interlineage divergence. If the divergence is high and diversity is low, it suggests that we have some positive selection going on, and vice versa. If, which is the case here, if the intralineage variation is pretty high or equal to interlineage divergence, we are probably facing a locus which is under neutral evolution, okay? And that will give us, in terms of FST values, it will give us a low FST value. Because FST gives us the idea of divergence, but this idea of divergence includes the comparison with also variation within one lineage, within actually the two lineages. So this FST is an indicator of the overall divergence, okay, and making a ratio. So it's a bit strange because this time it's the opposite ratio. Okay? It's not diversity divided by divergence, but it's the opposite. It's divergence between the lineages divided by intralineage genetic diversity. So if divergence is, let's say, low, or the genetic variability inside the lineage is high, we're going to end up with a low FST, a low broad sense FST, broad sense divergence. Vice versa now, which is a case that we are interested in, you have very low genetic variation within a lineage, but quite a high divergence between two lineages, which face again the case of a positive selection acting on this specific locus. And in this case, we're going to see a high FST. So basically, we can summarize it with a scheme. Low FST will be this typical situation. Basically, there is very high diversity within a lineage, and you see that both lineages share more or less the same alleles. So there is simply no divergence between them. And that is a typical case of having low FST. So probably two polymictic populations or two very close populations from the same lineage, something like that. Vice versa now, imagine that you have very high FSTs. That leads to what? That means very low diversity within a lineage. So it's suppose the case here, you see only one allele in lineage A and one allele in lineage B. And very high divergence because there is no shared allele between lineage A and lineage B. And that gives you a high FST. And this is the case if we take whatever gene under selection, we then assume that, okay, selection acting in lineage B, red allele will be selected and get fixed, while its selection is not acting. Same selection is not acting in lineage A, which means that the allele red cannot be simply selected and cannot be fixed. So high FST is a sign of positive selection, but again, high FST compared to the rest of the genome, okay? It's not just value itself, it's really comparing it to the rest of the genome. Because again, high FST could simply indicate very distant lineages, and each of these lineage having, for some reason, very low genetic diversity within them. So it's really, really important when you do that, you will do that in a practical, really to keep in mind that fine selection, we can only compare the gene site with the background of the genome. That's the only way. Otherwise, we are mixing demographic events with selective processes. And this is how FST would look like. Again, this is the exact same story. So if we look at here, okay, so this red selected site in human, then you would see a peak, a high FST here. And then, of course, high FST compared to the rest of the genome again. While FST in this case, in neutral case, you simply would see nothing, no peak. And again, why is this time it's it's a high peak, it's because it's doing the opposite. It's dividing the divergence by the diversity within lineage. Okay, so we expect a high value when there is selection happening. Okay, so same again, a little quiz for you. Again, same story. Think about it, pause the video, and put your answer as a comment in the timeline of the video so that the others can see. You can comment and you can discuss. The question first is, if we have a gene under positive selection in a population or lineage, what should you see? So should you see high genetic variation within this population plus low genetic divergence between this population and sister lineage? Or the opposite, low genetic variation within this population, high divergence between this population and the sister lineage? Should you see a low or a high Tag-MASD compared to the rest of the genome? And should you see a low or a high FST between population and its sister lineage? And again, compared to the rest of the genome. Okay, first one. Now, what about a gene under purifying selection? So same story, what should you see there? Low genetic variation within this population and low genetic divergence, or low genetic variation within population and high genetic divergence, or vice versa, high genetic variation, low genetic divergence. And Tag-MASD compared to the rest of the genome, is it high or is it low? And that's it. And then we will go for the last part after that.